Osmosis and tonicity are pretty important concepts to understand when you are trying to learn about what's going on inside the human body and the processes that happen there. So osmosis is the movement of water. It moves from a hypotonic to a hypertonic solution. And this is determined by the percentage of solute, how much stuff is dissolved in the water. So we're inside the body, we're talking about it like inside a cell versus outside of a cell. So, you know, bloodstream going past cells, you've got water in the bloodstream, you've got water inside the cells. How much stuff is dissolved in the water, which determines the hypotonic and the hypertonic. So you demonstrate this with an osmometer, which you guys will make with dialysis tubing. And you've got to understand tonicity. So this is what I'm talking about when I talk about how much stuff is in the water. It's tonicity. So tonicity it's a concept that you can only talk about as a comparison. It's like big and little. If I tell you that a tree is big, okay, that's fine. But if I don't, you can say big. Well, in your head, you may think it's big compared to another tree. So you're thinking like redwood compared to, you know, something like a dogwood that doesn't get as tall or as large. Or maybe you're thinking it's big compared to an ant. But if you're thinking big tree and I'm really talking about, you know, something else, you've got to have that comparison. So with big, you've also got to know it's bigger than something, or little, it's littler than something. So this tree could be big if you're talking about comparing it to an ant. Ant is little, tree is big. But that same tree could be considered little if I'm talking about the tree compared to the entire Stephen F. Austin State University campus. Now the tree is considered little. So you've got to have that comparison mindset in mind when you talk about tonicity, because you can't have a solution that's all by itself that is hypertonic or isotonic or hypotonic. It has to be compared to something else. Let me show you what I mean. In this first situation over here on the left, I have a hypertonic cell. That means there is a lot of solute dissolved inside the water inside the cell. It is hypertonic compared to the solution outside the cell because there's not as much stuff dissolved out there. So a lot of stuff dissolved inside the cell, not so much outside. This makes the cell hypertonic and it makes the solution outside the cell hypotonic. You got to have that comparison. So when you have this situation, water is always going to move, and it's just the water that moves. It is not the solute. It is not whatever is dissolved in it. And this could be, in the case of your experiment that you're doing, you're talking about sugar dissolved in water. So you've got sugar water, or in the case of a cell, you're talking about all the proteins and the fats, and well, fats don't really dissolve in water, but they're part of a cell and they're part of what's in there. But you're talking about all of these little pieces, the organelles, the proteins, the little nutrients that are floating around, you know, the little molecules of glucose or of oxygen. All of these things are dissolved in the water inside the cell. So when you've got a situation where you've got a hypertonic solution and a hypotonic solution with a membrane between them, the water is going to move from hypotonic to hypertonic. So it's in this situation, it's going to go from outside the cell to inside the cell. Now, if it's the same, if you look at this middle, you've got 1, 2, 3, 4, 5 little bits dissolved outside the cell, and 1, 2, 3, 4, 5 little bits dissolved inside the cell. It's the same. The water could move back and forth. If some moves into the cell, then some's going to move out of the cell, or if some moves out, then some will move back in. It just keeps this basic balance from everything. This last situation is the opposite of the first one, a hypotonic cell and a hypertonic solution. So this happens like when you get really dehydrated. You've got more water, a higher percentage of water inside your cell than you do in your blood. And so that can cause problems for your cells because the water will start to leave the cell and go into the bloodstream. This is more like if you've drunk a whole bunch of water and your kidneys can't process it fast enough to clear it out of your body, you're going to end up with the water going from your blood into your cell. So this is the opposite. You've got a hypotonic cell and a hypertonic solution. So remember, when you are answering the questions on your worksheets about the tonicity, you have to tell the tonicity of the cell compared to the outside solution. Look at which way the water moved, look at how much sugar is dissolved inside in the solution that's inside the tubing versus outside in the cup. And so you're talking about what is inside that tubing compared to what is in the cup. So this is a little bit more about tonicity. I know there's also explanation in your background information that is really good to read. It explains all of this too, but I just find that sometimes it helps to have this little visual to help me remember which way the water moves and why. The water is not trying to balance the amount of water, it's trying to balance the percentage of solute.